So I looked at Western medicine, and I specifically looked at how the specificity of Western medicine really affects as a patient how you're treated and how you interact with your physicians. So imagine that you wake up with a sore throat. We all have. You kind of look in the mirror. You try to point a flashlight back then, and some weird stuff growing on your tonsils. So you decide, okay, I'm gonna go to the emergency room and see what the doctor has to say about it. So you go to the emergency room and you start talking to the doctor, and this is kind of what's starting to go through the doctor's mind. Is it an immediate life threat? If their throat hurts, does it hurt so much that they can't breathe? Or does it hurt because it's swelling and they're not about to be able to breathe? So once they kind of more manage the symptoms of any immediate life threats, they're going to start looking at the pathology and what's actually wrong with you. And then within that, they're going to start looking at, is it a bacterial infection or a viral infection? So bacteria are just all over your body. They're inside. They're kind of what makes your digestive system works. Less than 1% of bacteria will actually make you sick. The other 99% of it is actually very beneficial to you, and you actually need it to be able to live. And unlike other types of cells, bacteria doesn't have any organelles outside of the ribosome. And what the ribosome does is it reads the DNA or the RNA. Bacteria don't have both. And it creates proteins and allows the cell to actually duplicate. And because of this, infectious bacteria multiply really quickly because kind of their only job is to kind of spread and multiply and just make sure that they survive. And then bacteria come in three main shapes: spiral, rod, and spherical. They don't really have that much of an effect on what the bacteria do. It just kind of depends on what kind of bacteria it is. So then viruses, they're not really considered a cell because they don't meet all the qualifications to be a living cell. They only meet some of them, which is kind of weird. So they have nucleic acid, so they have DNA or RNA, and then they're enclosed with a protein coating that actually kind of makes up their structure. But one thing that kind of makes them not so alive is that they need a host to multiply. So in this diagram you can see that the virus comes, it floats around, it lands on the cell, it injects the cell with the virus's DNA, and then the ribosome, which copies DNA and makes proteins, goes around and it scans the virus DNA instead of the DNA that the cell was actually meant to produce. And then the cell is tricked into producing more viruses that are then released in the body. And depending on the virus will either then kill the cell once the viruses are produced, or the virus will stay alive and just keep pumping out more and more viruses. So then you kind of have to start thinking about, okay, well, they're infected. What is their body already doing to try to fight it? So the first line of defense is going to be your mucus response. And your mucus response is just on your skin, in your nose, in your lungs, and it's basically just trying to filter out and just keep everything out so that you don't get infected. So that's why if there's pollen in the air and your body's ooh, we don't want that stuff in there, you'll start sneezing it out and you'll have a ton of mucus. Or if something gets in your lungs that your body doesn't want there, it will instinctively just be okay, I'm going to cough it out. I'm going to try to cough up mucus and try to pull all of that out with it. The second line of defense is the inflammatory response. And the inflammatory response happens when your body, if let's say you get a cut and your body's okay, I'm bleeding, it has something has breached the mucous membrane, it's going to release histamine in your body, which is going to kind of open up the capillaries in your skin, and it's going to allow more blood to flow in, and that causes swelling. And basically what that swelling does, if you have a cut, is it kind of helps close up the wound so that it can kind of start to mitigate how many different bacteria actually get in, so that the rest of your immune system is a little bit better of a chance. But that can also cause issues of if you eat a peanut and you're allergic to peanuts, your body will freak out and release a ton of histamine, and that can cause you to swell up, which is not great. And then the third line of defense is your immune response. So there are two parts to immune response, and I'm going to start with the innate immune system. So the innate immune system is kind of like the foot soldiers of your immune system. And they're made up of white blood cells. So the first kind of white blood cell is called the leukocyte. And leukocytes travel, actually all white blood cells are in a family of cells called phagocytes, and they travel through a process called phagocytosis, which is like in this picture. They go up, they travel through the veins, and when they get to where they're going, they have the vein open up just a little bit, and it actually squeezes through. So if this is the inside of the vein, it squeezes through the outside of your body, so it can actually leave the vein. So then leukocytes, the first kind, basically their only job is that they're like the bouncers. They're basically, if you get a splinter, they're going to go up to that and they're going to collect and they're going to try to actually just push it up. And when they collect, it is pus. So like if you get a zit or a splinter anywhere and you notice that like it gets pussy as it's like, as the splinter's starting to come out, that's all the leukocytes starting to come together to try to push it out. Next is neutrophils, which is actually what you see here. They also travel through phagocytosis. And basically what they do is they go around and they look for pathogens or infected cells. And when they find one, either if it's from cells around it calling for help or it being marked by a B cell, which I'll talk about in a minute, it goes up and it will latch onto it and it will actually kill that cell. So it'll actually destroy the bacteria cell. The thing about neutrophils is that they can only destroy one cell and then they die. So the macrophages is Greek for big eaters, and they're kind of more like bodyguards that stay in your gut, and they do the same job as neutrophils, but they don't travel through your body, through the bloodstream like neutrophils do. And when they attach to a pathogen, they can kill up.
You can.
They can eat up to 100 before they die. And they also do something where they will attach to the pathogen, they'll break it down, they'll take some of its proteins and kind of neutralize it, and then put it on their outside of their cell wall, and then T cells will come collect information about it, which I'll talk about more in a minute. And then natural killer cells are cells that travel through phagocytosis, and when cells are infected by a virus, they stop producing something called MHC1, which stands for the major histocompatibility complex, and then natural-born killer cells attach them, break down their cell walls, and kill them. And it's kind of an interesting thing, as it's a human cell killing another human cell. And then dendritic cells do the same thing as macrophages, but they're just on the skin. Okay, acquired immune system. This is when things get a little bit more complicated. So your acquired immune system is broken up into two parts. You have the T cells and the B cells. So the T cells, there are several subtypes of them, and they are helper T cells, and helper T cells kind of like organize the rest of your immune system to be able to go attack. So they will go up to either the dendritic cell or the macrophages that have displayed parts of this pathogen on their cell wall, and they'll go up to them and the macrophages will release a chemical called interleukin-1, and that kind of chemically transfers information to the helper T cells about what to look for in this pathogen, and then the helper T cell will release a chemical called interleukin-2, which basically just alerts the rest of the body that they need to start paying attention. And one thing that it can actually happen is something called CAPS, which is cryoprion-associated periodic syndrome, and that's kind of a family of illnesses where your body will just mistakenly release interleukin-2, and that gets your immune system really fired up and trying to find something that's wrong, and then that has it just starting to attack healthy cells and just starts it really hurting itself. And there are a lot of clinical trials going on of trying to figure out how to manage not producing interleukin-2 but still producing enough of it that your body can respond to when you're sick. So anyway, so the T cells helper T cells gather information, and then they, some of them just kind of go dormant and remember that information, and then others of them go up to B cells, and they basically program the B cells. So they go up to the B cells, they dock with them, and then they talk to the B cells about what to look for, and then that programs the B cell with these things called antigens, which are these little stems. And then once the B cell is programmed, it just kind of floats around the body until it runs into the pathogen, which you see here. And then when it runs into a pathogen, it starts reproducing like crazy. And it reproduces what are called plasma or effector cells, and they just pump out antibodies, 100,000 antibodies a minute. And then antibodies float around, and they're specific to each pathogen, and they attach to the pathogen, and then the neutrophils can actually sense, like kind of chemically tell where the antibodies are, and then they can go up and destroy those cells. The other ones just turn into kind of clones of these ones, and they're called memory B cells, and they both kind of remember about it, and then they're also then on patrol in your bloodstream as it's flowing around, making sure that if you get infected later on, your body can recognize it really quickly and take care of it before it becomes a problem. So memory T cells just remember information about the pathogen. And then cytotoxic T cells, so when a cell is infected by a virus or a bacteria, they stop producing certain chemicals, or they know like, oh, a virus has taken me over, I'm just producing more viruses, and they know they're a goner, and they release a chemical saying like, I'm a goner, come help me. And then cytotoxic T cells will come up, and they will also kill other human cells that have been infected. So maybe wondering like, why does this really matter for my presentation? And what that means is that if it's a bacteria infection, they're going to start to think like, okay, well, we can culture this bacteria. We can like swab the back of your throat. We can grow it on petri dishes like we did in class pretty much. And then they will, if it turns out to be a bacterial infection, they'll prescribe antibiotics, which I'll talk more about in a minute. And then for viruses, there's not a whole lot you can do once you're infected by a virus. So they might ask themselves like, okay, well, it kind of looks like influenza. Were they vaccinated for it? And if they were, it's a problem because it means that they were vaccinated for it. And then there's like a different strain that's now out that they can get infected by. And if they weren't vaccinated, then that's also a problem because now they're sick and they've been infected by this virus. Cool, so antibiotics are given after a bacterial infection has started. And there are two types of antibiotics. There's bacteriostatic, which basically just like stops the bacteria from reproducing, so it interrupts their protein creation or it messes with their DNA. And that basically just gives the immune system a chance to catch up and wipe out all the bacteria. And there's bactericidal, which actually just kill the bacteria and kind of take the place of the neutrophils. But one thing that can happen with antibiotics is that it kills all the bacteria in your body. And since only about 1% of bacteria is bad, that means that the 99% of bacteria is killed. And another big thing that we talked to people about when we went to a pharmaceutical company here in Boulder was that it's really important to try to like have narrow spectrum antibiotics, because if not, it does kill all the bacteria. But also, if even like one bacteria survives, it learns information about that antibiotic, and then it can grow back to be resistant to it. And the CDC estimate that in 2014, at least 2 million people became infected by bacteria-resistant antibiotics, which is like insane, and that number is just going to keep getting bigger as we keep using antibiotics. So then vaccines, they have to be given before someone is infected by it. And basically, it's just small doses of a weakened version of the pathogen so that those T cells can learn about it and then program the B cells to be on patrol to see if it has become infected. And some of the preventable illnesses, this is just a few, the CDC is a list of like 14, but are hepatitis B, rotavirus, diphtheria, influenza A and B, measles, hepatitis A, and poliovirus. So, and the other good news is that people are greedy, and they'd probably just like, drug companies would just pump out all sorts of weird stuff to try to get you to pay for it. So the FDA has created a process to make sure that the drugs are like actually work and they're vetted. So it starts with the discovery and development, and that's kind of usually done by either the CDC or just private companies that are trying to develop drugs. And then the protocol creation and protocol review kind of vary based on who's doing it. Basically, it's like they just have to come up with what the experiment is and what they're looking for, and then the protocol review is just kind of like vetting it to make sure it works. Something that happens that is not actually in this visual, but is right there, is preclinical trials. So that's looking at it in either animals or in petri dishes to make sure that like it actually is affecting what you're trying to affect, and it's not just like pumping weird stuff in the body. So then in the trials, phase 1 is just like really small doses of the medication to see what the side effects are, and just to like kind of see what the appropriate dosage is, and that's in healthy people. Phase 2 is kind of the same thing, but in sick people, so seeing if it has like any results in them. And if you pass phase 2, as you get into phase 3, you start getting into just like getting as many people on the drug as you can to see, just to collect a lot of data and make sure that it really works. And then the FDA reviews all your research and approves it. And then kind of for the life of the drug, the FDA will continue to kind of monitor it and make sure that you didn't change it without telling them. So, how does this affect how patient is treated? So, it can be nice when you go to the doctor if they can be like, hey, you're infected by this specific kind of bacteria and this is what's wrong with you. And that can be like really reassuring. If like, oh yeah, cool, like you can give me a drug, I'll be fine. But it is not great when you go and they're like, we don't really know what's going on with you. We're either gonna have to do more tests or like, we don't think you're gonna die, come back if you start feeling way worse. And also the specificity means that like, we understand how infections work down to like the cellular level. And this means that like no one doctor fully understands it. Like there are a little over 8,000 podiatrists in the US. So that means that there are like 8,000 doctors who have studied just to work from like your ankle down for like 10 years. And like this can be frustrating if like your entire leg was destroyed because then you need a whole team of doctors to help you. But at the same time, that can be really nice because you know that your foot is going to get repaired to like the best possible state it can be because this person spent only 10 years. But one of the bigger issues is that when you see it as a cellular level, you start being seen almost as like a living anatomy book. And it's like, I know I've definitely had experiences with this at the doctor where it's like, they're not really like looking at the person in front of them, they're looking at like the bacteria in the back of your throat, and they're kind of trying to manage that instead of managing the whole patient. And it's also, I'll talk about that in a minute. Cool, so then this kind of gets into like, how does this shape just kind of a worldview? And I think that it focuses a lot, like nobody goes to the hospital when they're healthy. And I think that that kind of says a lot about the Western culture as a whole, that like, there's this thing goes around and it's like, don't fix what's not broken, but maybe it's not broken just because you're not looking at it. So, and you're not doing anything to prevent it because it's not broken yet, even though it's going to break at some point in the future. And like in Western medicine, there's no real focus on prevention. It's just like, if you're sick, go to the doctor. If you're not sick, don't go to the doctor. And also that like whole idea of like focusing on the pathology and focusing what's wrong kind of transfers over to like the news. I don't know if you guys have watched the news recently, but it's always just like, this mass shooting happened, this cop did this in this city, this other bad thing happened, and very rarely it's like, this is what people are doing to prevent this, or this is what, like, some good things that happened. And then also the specificity of like what's wrong, kind of like, I don't really know how to describe it, but it's like when, like, we want to know about details in our lives, but I know like I'm way more interested in details in my lives about things that like affect me negatively, so I can like try to get ahead of them. And like just details in general, it's like as a culture we're so like, we want to know like every single thing about that celebrity or every single thing about this other person, when really we should be focused more intrinsically and focused on like, what can I do to make myself the best person I can. So that's all I got. I'll get off my soapbox.
How long was that?
18. Any questions? We don't have time.
You don't need to know this at all, but do you have any idea what the percentage of drugs to get approved by the FDA is?
I don't know the percentage. I think I saw something about like, for like every 100 drugs that starts, only one gets approved. I wouldn't quote me on that, but I kind of vaguely remember that.
So I know you're very interested in health and you've been in the for training and all that. How do you see these connections come together within your
It's an interesting question. I think that like a lot of the medicine really came together of just like I like to know why things are happening a lot of the time. So like I got sick the other day or like a few weeks ago, and I was like kind of interesting. I started noticing in myself like, oh, like this is why I feel bad, and this is like probably what's happening, or like if like and then trying to connect this to my woofer, which happened a lot. It was like thinking about like, oh, well, if they present with like these signs and symptoms, or if they get cut and like pus is coming out, like it's like this is what's happening. So I think that that was just kind of like an interesting connection that I've been making in my brain.
Awesome. Great job.
Good. All right.